For this presentation, I assume that we know the minimum basics about the structure of DNA, that DNA is a macromolecule, or we can say a biomacromolecule. Therefore, synthesis of DNA is all about molecular interactions. Here, we are going to focus not so much on the description of the phenomena in terms of the molecules involved, but on the structural aspects of DNA synthesis. The lecture is based essentially on the book Fundamentals of Molecular Structural Biology. Now, synthesis of DNA is required in any of these three cellular processes. Number 1, replication. Number 2, repair. And number 3, translesion synthesis. In replication, two identical replicas are produced from an original DNA duplex. Here, we see the ongoing activities based on one of the two original strands. Deoxyribonucleotides, or DNTPs, which are the building blocks, are added one after another to the growing complementary strand. In repair, damaged or altered nucleotide residues in the DNA are restored to their normal state by DNA synthesis. A portion of the strand containing the damage is removed by repair enzymes. Thus, a gap is created in the duplex. This gap is subsequently filled up by new DNA synthesis. In translesion synthesis, a damage or lesion in one strand of the DNA is bypassed by an error-prone incorporation of nucleotides in the opposite strand. Now, let us say there is an oxoguanine, shown in red, instead of a normal deoxyguanine in an original strand of the DNA. Now, normal DNA synthesis on the growing complementary strand would stop at the abnormality or lesion, as shown by the red dot. However, translesion synthesis would incorporate an incorrect nucleotide at the site and let normal synthesis continue downstream. Let us look at the basic chemistry of DNA synthesis. We have here the growing portion of a polynucleotide chain, the 3' hydroxyl and a deoxynucleoside triphosphate, that is a DNTP. Now, a proton is transferred from the 3' oxygen to an oxygen of the alpha phosphate. The transfer is shown here. The activated 3' oxygen is now competent for nucleophilic attack on the alpha phosphate of the DNTP. As a consequence, the bond between alpha and beta phosphates is broken, and a pyrophosphate is ejected. This pyrophosphate is subsequently hydrolyzed by an enzyme pyrophosphatase into two inorganic phosphates. On the other side, a new phosphodiester bond is formed, and a DNMP is added to the growing chain, which is longer by one residue. The entire process involves a free energy of 8 kilocalories per mole, which is quite favorable for the reaction. So, the basic chemistry of DNA synthesis says that if we have a growing polynucleotide chain with a 3' hydroxyl end, in an environment of dNTPs, then addition of a dNMP to the growing end is possible under favorable conditions. And this dNTP can be either of dATP, dCTP, dGTP or dTTP. The basic chemistry is same for all four. Therefore, the obvious question would be, is the nucleotide addition random? The answer is no, it is not. The specific direction is provided by the template DNA. The growing chain should act as a primer, and the primer is required to base pair well with the template and form a specific structural arrangement called the primer template junction or the PT junction. So we see that nucleotide addition is not random but template directed. The component requirements for the nucleotide addition or polymerization reaction are thus two substrates, the PT junction and 4 dNTPs. We also have the requirement for magnesium ions. All these components need to be assembled in a molecular machinery called the DNA polymerase. A large number of DNA template dependent DNA polymerases have been discovered in a variety of organisms spread around all three domains of life: archaea, bacteria, and eukarya. Based on their amino acid sequence homology and crystal structures, the DNA-dependent DNA polymerases have been grouped into 5 families. Here is a representative from each family: E. coli Pol1 clino, family A, with replicative as well as repair functions; bacteriophage RB69 GP43, family B; Geobacillus Pol C, belonging to family C, required for DNA replication; human Pol beta, family X, having repair function; and Sulfolobus DBH, belonging to family Y, involved in transclusion DNA synthesis. For productive interaction of the molecular components participating in DNA synthesis, the DNA polymerase, whether replicative, involved in repair, or transclusion synthesis, is required to stably hold together the double-stranded DNA, including the PT junction, dNTPs, and magnesium ions. All the polymerases therefore share a cleft-like architecture. There are 3 common subdomains: thumb, palm, and fingers. The palm subdomain is at the base, while the thumb and fingers form the walls on the 2 sides. The thumb properly positions the duplex DNA and facilitates processivity and translocation. The fingers subdomain is involved in interaction with the incoming dNTP and the template base to which it is paired. The palm is formed by alpha helices stacked against a beta sheet. Here are the palm subdomains of the DNA polymerases we have seen in the previous slide. This subdomain carries the active site acidic residues required for the catalysis of nucleotide addition or phosphoryl transfer reaction. The cleft walls, on the other hand, are dominated by basic or positively charged amino acid residues. Therefore, as would be expected, the DNA polymerases belonging to different families follow a common strategy for nucleotide incorporation. Polymerization begins with the binding of a primer template PT to the unliganded enzyme, the polymerase denoted by E. An enzyme primer template EPT complex is formed. This is step 1. Next, in step 2, a DNTP coordinated by a magnesium ion binds to the EPT complex to form an EPT DNTP ternary complex. Selection of this DNTP from 4 kinds is directed by Watson-Crick hydrogen bonding with the template base called the coding base, which is just beyond the 3' end of the primer. This ternary complex undergoes a conformational change to be activated. The activated complex E'-PT DNTP is competent to undertake the nucleotide addition chemistry. In the next step, the 3' hydroxyl of the primer activated by the interaction with a second magnesium ion attacks the alpha phosphate of the DNTP and forms a phosphodiester bond. The pyrophosphate is hydrolyzed into 2 inorganic phosphates by a pyrophosphatase activity. The first step in the polymerization process, that is binding of the primer template PT to the enzyme, requires geometric and electrostatic compatibility between the 2 components. In the case of E. coli clino, the active site cleft is about 25 angstrom wide and 35 to 40 angstrom deep. Now, the diameter of B form of DNA is about 24 angstrom, so it can be easily accommodated into the cleft. On the other hand, we have already seen that the cleft walls are dominated by basic or positively charged amino acid residues. The DNA backbone is negative due to the phosphates. So, the electrostatics also helps in the binding. As a result of PT binding to the polymerase, structural changes occur mostly in the thumb subdomain. The thumb opens up initially but quickly rotates in the opposite direction to bring the residues in a helix-to-helix motif, which is at the tip of the thumb closer to the DNA. Thus, a cylindrical formation wraps the DNA and the 3' end of the primer is brought near the polymerase active site. Additionally, in the cases of DNA polymerases, such as those from Thermus aquaticus and E. coli, a tyrosine residue enters into the stacking arrangement of the template bases and holds the first base pair of the PT duplex in the active site. Next, A DNTP binds to the EPT complex to form the EPT-DNTP ternary complex. The binding is helped by electrostatic and stacking interactions. Here we see the example of Taq DNA polymerase, Taq1. The DNTP binds to the N-terminal end of a helix called the O helix in the fingers subdomain. There are electrostatic interactions between the triphosphate group of the DNTP with the positively charged side chains of an arginine and a lysine, and stacking interaction with phenylalanine. However, in this ternary complex, which is an open complex, the nucleotide is still not close enough to the active site. It has to wait for a large conformational change in the fingers subdomain. The necessary conformational change does occur, and the EPT-DNTP open complex is immediately converted to the activated E'-PT-DNTP closed complex. There is considerable rotation, and the O helix moves closer to the active site constituted by the 3 carboxylate residues. The tyrosine of the O helix, whose aromatic ring was earlier stacked into the first base pair of the BT duplex, moves away from the stacking arrangement. The template base positions itself to base pair with the correct incoming nucleotide. The transition organizes all the components of the active site topologically and geometrically so that the polymerase is able to proceed to the chemical step. In all DNA polymerases, the phosphoryl transfer reaction takes place by a 2-metal ion catalytic mechanism, and in most cases the metal ion is magnesium. 2 magnesium ions are coordinated in the active site by the phosphates of an approaching nucleotide, carboxylate side chains of aspartate residues, and water molecules. This enables a magnesium ion to interact with the 3' hydroxyl of the primer and facilitate the proton transfer that we have seen in an earlier slide. And as it has been already described, the activated 3' oxygen attracts the alpha phosphate, resulting in the formation of a phosphodiester bond.